Good evening, esteemed members of the profession. Thank you for joining us today. If you can hear me, just send a message on the chat. Let us know which branch you're joining us from. Dr. Sorej, Dr. Albert, can you hear me? Right, thank you so much. I believe you can hear me. Dr. Samson Juma from South Rift. Welcome. Dr. Rama, Dr. Daniel, thank you for your response. Karibuni sana. Thank you for your response. I see most of branches are represented. Asanteni sana for joining. Meanwhile, you can be reminding your colleagues to join. Thank you for your responses. So today's webinar is on biologics and biosimilars. And it is sponsored by Sanofi. And our speaker today is Dr. Imran Manji, who is a public health pharmacist. And just before we get started, I'd like to take you through a few ground rules and announcements. And first, it's to thank you for joining this session. And to remind you that you'll be muted during the entire course of this session. And if you have any questions, kindly utilize the Q&A tab at the bottom of your window. And questions will be responded to by the presenters on the Q&A tab and verbally at the end of the session. For all the PSK members, if you have not done so, kindly subscribe on the PPB portal. Also to note that this session is being recorded and it will be made available on the YouTube page. So just to help you find this event easier on the portal, just search for the word biologics or biosimilars, and you'll be able to get this event. So in fact, before we start, see we have 1 more minute to 7:10. You could kindly log into your PPB portal and just sign in and make sure you subscribe to this event on the portal. Kindly note, there are 2 events. There was one that was done by HOPAC, and this is now done by PSK. So make sure you subscribe to the one that has been written, Pharmaceutical Society of Kenya. So the agenda for today is we'll have the presentation by Dr. Imran Manji. Then thereafter, we'll have a presentation by a representative from Sanofi Group Ethica. That is Maurice Tembo. Then thereafter, we'll have our Q&A and then close the session. So at this point, I'd like to welcome our speaker for today, Dr. Imran Manji. Dr. Imran, if you can hear me.
Yes, good evening.
Good evening.
I hope you can hear and see me.
Yes, we can.
Great. I'll go ahead and share my slides.
That's okay.
Please tell me if you can see my slides.
Yes, we can see. You can just put them on full screen mode.
Just trying to do that. There you go. Great. I can get started?
I don't know if it's still on full screen mode on your end, because on mine it's still showing it's not full screen.
Is it not visible?
No, it's visible, Daktari. It's not on the slideshow. You can just touch on the slideshow.
Can you hear me?
Yes, we can hear you clearly.
Can you see my screen now as a full screen?
Yes.
Yes.
From my end, not yet. Slideshow. But I can see your screen. But they are yet to go on slideshow.
Sorry, I can't hear you. Maybe my internet connection. 1 second.
Yeah. I think there's a problem with the internet connection.
I'll just turn off my video. I think that's probably part of the
Okay.
problem.
Okay. Then you can just try put it on slideshow again.
Okay, just a second. I'll just stop share. Is that okay now?
Yes, it's okay.
Yes, it's okay. Very fine.
Great. I'll go ahead. So, good evening again, and sorry for the hiccups. Thanks for the introduction. And first of all, thank you, PSK and Sanofi, for inviting me to do this presentation. My name is Dr. Imran Manji. I am a pharmacist. I work at Moi Teaching and Referral Hospital. I think I know many of you who have joined in, so it's a pleasure to be here. I'll get right into it, just to keep time. So we're talking about biologics and biosimilars. And for today's presentation, basically this is what we're going to go through. So we'll talk about biologics, what they are, how do they compare with the drugs that we know, the chemical drugs, what's their manufacturing process. Then we'll move on to biosimilars. Again, the same thing. What are biosimilars? And then we'll compare with generics and how do we get regulatory approval for biosimilars. We'll move on to talking about something called non-comparable biologics, and then the general regulation for biological drugs and biosimilars. And then finally, we'll say something about interchangeability. But before we get started, I have a little case for you, and I would like you to give me your thoughts on the chat, so we can go through the case. So a patient in your hospital is being managed for a rare cancer with immune therapy. The drug is a monoclonal antibody given as a weekly subcutaneous injection. It has been prescribed for a period of 3 months, and the patient has been taking the drug for at least 6 weeks. Today, the patient comes up to you as the pharmacist and says that she is unable to afford further treatment because of the high cost of the drug and is requesting for an alternative. You are aware that there is a quote-unquote generic of this drug in the market that would be much more affordable for this patient, but you have no further information about it other than where to source it from. So what do you do? You have, these are the options that you, So if you can put on the chat, do you just go ahead and purchase the alternative and dispense to the patient? B, speak to the oncologist and recommend substituting with the generic. C, request for more product information from the distributor of the generic, including registration with PPB and international regulatory authorities before deciding to substitute. D, counsel the patient that the product she's taking is the only option she has, as the quality of the generic is unknown. E, discuss alternative and more affordable therapies with the oncologist. So give you guys a minute to respond. I'm seeing a lot of E's, but wow, okay, that's a lot of responses. I'll just quickly sample. I'm seeing majority E, a lot of C's, no other. C and E is the most common. I'm seeing a B, speak to the oncologist and recommend substituting. I'm seeing, but mostly E's and C's, B's. Great. I think we'll stop it there, but thank you for your responses. I will not give you the right answer right now, but as we go along. So So thanks for interacting. We'll move forward with the presentation. So what are biologics? If you look at the definition, basically these are very large molecules. They're complex and they're produced through biotechnology. That's where the word biologics comes, or you even call them biological drugs or biological medicines. And they're produced in a living system, such as a microorganism or a plant cell or an animal cell. And some of the key things to remember, first of all, the manufacturing process is very complex, and we will see if you compare to when we compare to other drugs, that the manufacturing process is a lot more complicated. Examples include vaccines. I think these are items that we use almost every day. So blood and blood products, monoclonal antibodies, which was there in the question we saw earlier, gene therapies, proteins, you name it, yeah, insulin. We use these things every day. So low molecular weight heparin is another one. And they have a variety of indications, as you know, cancer, diabetes, autoimmune disorders. So many of these conditions require biological products. And an interesting fact is that they represent 20% of global pharmaceutical sales. Basically, that means that 1 in every 5 products that you are handling or dispensing or prescribing is a biological product. So that's why it's so important to understand what these things are and how they are manufactured, and how can we, if we have to substitute, how do we substitute? So this is just an illustration of some of the examples of common biological products that we have. You're seeing there's things like gene therapy, vaccines. Nowadays, immune therapy is the way to go in terms of in cancer. We are seeing so many monoclonal antibodies coming into the market and all of these are essentially biological products. So how do you compare chemical drugs versus biologicals? This is just in terms of size and complexity. On the left, you'll see aspirin, which is one of the smaller molecules that we know. But even if you look at the most complex chemically synthesized drugs, an example I try and remember from my pharm chem back in school many years ago, like amphotericin B, for example. It's so complex. If you try and remember the structure, you won't remember it. At least I don't remember it. But still, in comparison to biological products or biological drugs like a peptide or a protein, they are not as complex. If you remember biochemistry, you know how you have proteins as basically a sequence of amino acids. And when you compare, there's different structures, right? There's the primary structure, which is just the single chain amino acid. Then you have secondary, tertiary, quaternary. And the way these proteins are even in a spatial sense organized makes a big difference in the way they work in the body. So you can imagine how complicated it would be even just to manufacture these. And so just a comparison between the two types of drugs that we're talking about. We talked about the size and the complexity. Biological products are much more complex and they're heterogeneous. They're produced by living cells, whereas chemical drugs, we synthesize in the lab or we extract from plants. The process for producing biological products is highly process dependent. Basically, that means that if you change anything in that process, you may not get the same product at the end of that manufacturing process. Whereas with synthetic drugs, you might use so many different types of processes, but get to the same product, and you will eventually get to that same product. It's very different for biologicals. And then even the process of manufacturing is simpler for chemical drugs, and it's easy to reproduce. Whereas with the biological products, it's highly complex. There's all these conditions that you need, optimal conditions of temperature, pH, oxygen for the microorganisms that you're using, the nutrients. And then the other thing is that most of these biologics are immunogenic. That basically means that they can cause an immune reaction when introduced in a living system. And you know that proteins do that, whereas most synthetic drugs are non-immunogenic. And then when it comes to the storage conditions, chemical drugs are generally stable. Most of them don't require things like refrigeration, but biological products are mostly, not all, but mostly, they require special storage conditions. So that means you have to think about how are you going to transport these if they require refrigeration, cold, the cold chain. All of us know about that. So we need to, in biological products, it's a little more complicated, as you can see. Just a little bit about the manufacturing process. If you're using, an example is, say, insulin, you're going to use, there's a special cell line that you would use for manufacturing insulin, which starts with placing the DNA into your, into your target organism. Then you clone it with a different vector, transfer it to your host cell, and then when you start mass production. So you'll have your growth media, and then you start, like, you put them in bioreactors, and eventually you start extracting now the final protein that you're, for example, if in this case, if it's insulin, you will extract it and then purify it, and then you'll have the final purified drug. Now, all along this process, there are so many changes that can happen and so many factors that can influence the process, that at the end of the day, at the end of the whole process, you may not get what you're looking for, or the purity of that product might not be as per the standard. So you can imagine, like, if you have something changes in the cell line or in the media, for example, you will have changes in that whole process. And so, as I mentioned, you have variations, and because this is a biological product, remember, we're dealing with microorganisms, plant cells, animal cells. There can be a lot of factors that come into that, and we don't need to go into all of that detail. But at the end of the day, you could have some differences. Yeah, some slight differences are acceptable, and there are certain normal limits that the regulators place, like FDA has certain limits for every product that you would have for the final product, and this is based on the originator reference product. That these are the slight differences which are acceptable, and then they assess within product variations and what is the manufacturer's strategy to limit these variations. So you don't, while some are acceptable, but you need to limit them during the process. And then the other thing that's important in this process is that process can change. When you put new raw materials, the process might change. If you change the manufacturing site, again, what you get at the end product might be different. The cell line will affect. So all of these require approval at the FDA level, if you're talking about FDA, when you're changing these processes. So, they're very specific about these biological products, just because slight changes can lead to big impact down the line. So then what are biosimilars? So biologicals they are the originator reference product. Biosimilars are now the same biological drugs which are designed, it's in the name, that are designed to be highly similar to the existing marketed biologics. So for as far as FDA is concerned, biosimilarity is that this product is highly similar to the product, notwithstanding to the reference product, notwithstanding minor differences in clinically inactive components. So the primary amino acid chain must be the same, but other inactive components, a protein is quite huge. So some changes in the structure or the function of that protein are acceptable, but there should be no clinically meaningful differences between the reference product and the biosimilar. And that is in terms of safety, purity, and potency. So that we're talking about efficacy, when it's clinical efficacy and safety, and then of course purity, when you see the final product, it needs to be at the same level of purity as the reference product. So are biosimilars the same as generics? The answer is no. Generics would be identical to the chemical product, if you see here. And we know the process is the chemical synthesis. We already talked about that. Whereas with biosimilar, there are so many different steps along the way that could change that you will not get an identical product, even as much as you try. They are highly similar. So there's certain reference ranges that they have to meet to be able to say that you are biosimilar, and we look at that. And with, as far as generics is concerned, that process is easily reproducible, as I said. But biosimilars, we cannot even call them generics, because that's a totally different terminology. Just again, a take on how long it takes to develop biosimilars versus generics. You can see the time and cost for generics is much lower in comparison to biosimilars, because they are, we're almost saying they're similar to biological originator products because of the amount of investment and time that goes into getting these biosimilar products. And then just comparing biologics versus biosimilars, you can see even the cost and the time is not so different. And I think there's a push now to say, can we recognize biosimilars as originals across the globe, because of the amount of investment that goes into just developing these biosimilar products. And let me just say, so in terms of regulatory approval, so the FDA uses something called a stepwise approach, whereby they look at so many different aspects, and they call it the totality of the evidence. This is essentially the different evidences that are that would lead to them saying this product is a biosimilar and this one is not. And so it's very, very intense as to what they assess. So you look at now, first of all, the structural and functional characterization. We talked about that primary amino acid sequence. It must be identical. And then they'll do a non-clinical, you look at the non-clinical evaluation. So all of this data has to be submitted by the applicant. So you're talking about animal studies, you're talking about immunogenicity, we're talking about pharmacokinetic and pharmacodynamic data, and then comparative clinical studies. You remember, we have to show that there's no meaningful clinical difference between the two. So what are the safety studies? What are the efficacy studies? And until all of these have been met, these products will not get FDA approval. And it's the same thing in EMA, WHO. And then after all of that, once the product has been approved, the FDA and the manufacturer have to do robust post-marketing safety monitoring. Basically, that means that they have to go back, survey these products, and be very vigilant about, are there any new side effects? Are there any, is it more immunogenic? Is it affecting, are there efficacy issues? So all of these need to be met for these products to continue being in the market. And so what do we mean when we say highly similar? We talked about the amino acid sequence. Then there's all these higher order, structures of proteins. If you remember your biochemistry, how they're attached to the target receptors, what is their pharmacokinetics, solubility. We talked about immunogenicity. All these things have to be met before now they can be classified as highly similar. And you can see in this graph here, you need to meet, it may not be exact as the reference product, but it has to be within a very, narrow range. So we have something called non-comparable biologics, or copies, and these are not biosimilars. So when we say NCBs, these are biological products that they do not meet the requirements of similarity to the original product, and they have not been through the strict requirements. We talked about all of the comparability and the studies required by the relevant bodies like FDA, WHO. So if they do not meet these, then they cannot be classified as biosimilars. Now, why are these products in the market? Some of them are old. They've been licensed previously when there wasn't really all of these stringent requirements. Others have been manufactured because they have alternative indications that don't require such stringent comparative studies for them to be recognized as biosimilars. So until they meet the requirements, then the FDA doesn't recognize them as biosimilar. And these are some of the comparisons between non-comparable biologics and then the originator and biosimilar. So you see, most of these have not been evaluated for the quality, for efficacy, safety, so that means you can't extrapolate to the indications that they may have. So if it's, say, a monoclonal antibody, are you sure that you can extrapolate that indication that is for the reference product to this product? Similarly, they might have different, the processes are unknown. You can see a lot of unknown. That's because these products have been registered or approved without having all of these studies done on them, or they may have been done, but they've not been published, or they've not been recognized by the regulatory authorities. Same thing with safety, immunogenicity, clinical efficacy is what we talked about. The post-marketing we talked about. After they've been approved, what are the risks? We don't know what the risks for these products are, because none of that is out in the public realm. So in Kenya, what are the regulations that we have for biological products and biosimilars? There are two guideline documents. They're fairly recent. One is the submission of documentation for registration on biotherapeutic products. So now these are for the reference products, and then another one for similar biotherapeutic products or biosimilars. And so if you meet the requirements on this, then you will be registered through the Pharmacy and Poisons Board. But as you can see, if it was 2022, then it's not clear what was happening before these guidelines were in circulation. So lastly, we have something about interchangeability. What does interchangeability mean? First of all, we're used to substitution when patients come to the pharmacy, where if a particular brand is not available or a particular product is not available, the pharmacist has the clinical knowledge and ability to substitute with an alternative which is similar in maybe a generic or something like that. With biologicals, interchangeability is very, very there's a lot of nuance to it, and according to the FDA, if you need to meet the standard for interchangeability, the product must demonstrate biosimilarity. We've talked about that. It must produce the same clinical result as the reference product. We have also talked about that. And then the risk in terms of safety or diminished efficacy, between switching from the reference product and the biosimilar, must not be greater than when using the reference product. And then interchangeable products may be substituted for the reference product without the intervention of a prescribing healthcare provider. So within biosimilars, if a product has been classified by FDA as being able to be interchanged with the reference product, at that point, the pharmacist is able to substitute without referring back to the prescriber. But if not, then you would still have to discuss with the healthcare providers. So that's essentially interchangeability. And as I've been quite brief, I can see. So in summary, we talked about biologics, which are highly complex molecules. They've been produced with biotechnology. We saw the process. We've seen how they're used. And then for a product to be biosimilar, it must meet the requirements of safety, purity, and potency. And so, and if you don't meet those criteria and have no, and there are clinically meaningful differences, then you don't qualify to be a biosimilar. And you've seen the regulatory process for that through most of the stringent regulatory authorities around the world use the same process. And then we've said that non-comparable biologics are not biosimilars, and they cannot be used interchangeably with the reference drugs. Now, going back to our case from earlier on, it's actually as you can see from the presentation, it's a bit complicated because number one, you're seeing the patient is not able to afford the reference product. You are seeing that there's a so-called generic. We've talked about this, that you can't call them generics, number one. And so what would you do? I think in this case, there are multiple answers. And the way I looked at it was, you could start with C, get more information. Is this product registered with PPB? Is it in line with the requirements of international regulatory authorities like the FDA, EMA? And then if it isn't, if it is, then maybe you could be fairly confident, say, yeah, we can substitute. If it isn't, then you would then talk about the D option, because she may only have that option because we don't know the quality and we don't know the history of that so-called non-comparable biologic. And then if she can't afford it, then I guess you're stuck with the looking at alternative therapies, which may mean not something as effective at whatever she's taking. Of course, this is it's not a real drug, so we would have to go back and discuss. There, I think we can stop there. Hazran, I'm giving it back to you.
All right. Thank you so much, Dr. Imran, for that insightful presentation. So at this point, I'd like to welcome our speaker from Sanofi, Maurice. Thereafter, we'll have the Q&A session. So meanwhile, for our members, you could kindly, if you have any questions, use the Q&A, and the questions will be responded to at the end of the session. Maurice, you're welcome.
Okay, I'm sharing my screen. Kindly confirm, Dr. if you can see my screen.
Yes, yes.
Okay, my name is Maurice Tembo. I work for Sanofi. I think I've seen a lot of people who I'm familiar with logged in. And I'm going to talk about one of the biologic products from Sanofi that has become a household product in this country. I think Clexane is present nearly in every corner of this country. And just at the onset, I would want to ask you to think about whether do we have a biosimilar for Clexane and oxaparin sodium in this country. That's something I want you to digest at the backside of your mind. If that is so, then, or if it is not, then what of this product that we have in the country? I think from Daktari's presentation, it's quite clear that if you follow, you could easily tell where this other products belongs. And to get us started, I'll look at VTE, where Clexane has the therapeutic role. So yeah, I'm going to briefly look at the burden of VTE and why we should speak about this low molecular weight heparin, and also try to look at the differences between low molecular weight heparin. The issue of interchangeability has been mentioned. I won't emphasize so much on it, but I'll touch on it. Then we will talk of Clexane in terms of its quality, in terms of its clinical evidence, and in terms of its experience, not just nationally here in Kenya, but you'll note that this is a global product. And if you are somebody who travels, then you probably will have come up across this product in other jurisdictions or in other countries. So to get us started, let's look at the burden of VTE. Why are we having these talks on biosimilarity, low molecular weight heparin, Clexane, and all that? And there's this study that was called ENDORSE study. It has a very complicated spelling, but basically it was an observational study that went to look at the issue of venous thromboembolism in our hospital and hospitalized patients. And according to ENDORSE, pulmonary embolism accounts for 5 to 10% of deaths in hospitalized patients. And this makes VTE the most common preventable cause of hospital death. I want you to highlight the word preventable, meaning this is something that could be prevented. Patients don't have to die out of it. And in addition to these deaths that we are talking about in quote, VTE is also associated with some chronic complications like post-thrombotic syndrome and chronic thromboembolic pulmonary hypertension. And this condition actually increase the cost of management of this condition and even the morbidity that is associated with it. So this is why VTE becomes really a burden, and we really have to talk about it. Another study that looked at the burden of VTE is called VITA or VITAE or VITA. VITAE is the arm that was done in Europe, and VITA was the arm that was done in USA. And according to VITA or VITAE study, that's due to VTE in Europe. It was done across 5 years. I just put this in summary. You can find this online and see the detail of VITA or VITAE study. And it demonstrated that deaths due to VTE was more than 500,000. And it exceeds combined deaths due to AIDS, breast cancer, prostate cancer, and even transport accidents. And you can see the numbers. So you can see this is like a global crisis that needs to be attended to. Now, back to ENDORSE study, there are some little details on ENDORSE because ENDORSE also looked at hospitalized patients. And ENDORSE primary objective was to look at out of the patients who were admitted, let me use that word in the hospital, how many of them were at risk of VTE? And out of these patients who were at risk of VTE, how many of them were put on prophylaxis to prevent these VTE problems? And then those put the risk of VTE in hospitalized patient or admitted patient at 52%. Meaning that if you have a capacity of, let's say, 300 bed capacity in an hospital and it is full, like I know most of our government referral facility, then if there are 300 patients, half of them, 150 patients will be at risk of VTE. And according to ENDORSE, out of these 52, only 50% were put on appropriate prophylaxis. You can look at the numbers on the right. We had 35,000 patients at risk of VTE and only 17,000 got prophylaxis. Try to bring this home at your facility where probably you practice and see that what is the gap, what is the level of practice to ensure that this VTE is prevented. These ENDORSE looked at 35,000 patients at risk and only 17,000 were put on prophylaxis. I dare say that some of these patients probably are not with us. And those also was divided on surgical and medical wing. And in the surgical wing, or the surgical arm, the risk was put at 64%. So when you look at the surgical ward, the risk goes up. 6 out of 10 of the patients there are at risk of VTE, and you need to be vigilant. And for the medical arm, for 42%. So we are talking of 4 out of 10 patients in your wards are at risk of VTE. And according to ENDOS, for the 30,000 surgical patients, only 11,000 got prophylaxis. So you can see the gap between the patients who are at risk and the patients who are put on prophylaxis. And for medical patients, look at 37,000 patients. Of course, medical patients tend to be a little higher, but according to ENDOS, the numbers was nearly the same. And you can see medical patients, out of 37,000, only 6,000 were put on prophylaxis. I think if you look at this back home, then you'll realize that this reflects the scenario we have, because even here in Kenya, we see that most surgeons are more sensitive to the issue of VTE than if you compare it to the physicians. And it is really demonstrated even in this study that a lot of medical patients who should be on prophylaxis are not being put on prophylaxis. So this was what ENDOS set to discover. And the call on this was that healthcare providers should be vigilant, and the prophylaxis, the risk assessment and prophylaxis should be improved. So it recommended that a comprehensive approach to optimize VTE management should be there. And we should have VTE awareness and put systematic risk assessment in hospitals and do appropriate prophylaxis. And this is deemed to improve patient safety in the hospital. So this is what we have been trying to really sensitize healthcare provider on this, that we help our hospital become VTE safety zone. A lot of patients will come or might come with other complications, other medical issues, but they end up that what makes them lose their life is not even the disease, but just this venous thromboembolism. So we are calling on you people to be more vigilant and improve on the prophylaxis and appropriate risk assessment and appropriate prophylaxis. So back to Clexane, which I want to really discuss with you briefly as one of the agents that mitigates the problem of VTE. And to get us started on it is the mode of action of Clexane. What does Clexane do? I believe this is a familiar product for those who are in the hospital settings. I know we have a lot of people who are also in the community, but I'm dealing with pharmacists here. So I'm pretty sure that everybody knows about Clexane. And this is how Clexane works. Sometimes we are asked whether Clexane can lyse the clot, but it's good to know that Clexane doesn't do that. It works on the coagulation cascade to slow down the coagulation process by inhibiting factor 10A, goes ahead to inhibit factor 2A, and this happens in the ratio of 3:1. So there's a lot of inhibition of factor 10A compared to factor 2A. And this is a bit significant because when you are trying to slow down the coagulation process, you also don't want to cause bleeding. And this is why Clexane has a clear balance between, preventing the clot formation and hardly causing bleeding on patients. Now, back to on clinical evidence, why Clexane? Clexane has a broad range of clinical evidence and indication, both in prophylaxis and in treatment to the management of acute coronary syndrome. And if you look at this diagram, you can see this can summarize the whole indication of Clexane, both in prophylaxis and in treatment. And from 1987, there was a discovery of DVT prophylaxis in hip replacement surgery and knee replacement surgery back then. And in 97, there was a clinical evidence on DVT prophylaxis in abdominal surgery. And if you come to 98, you will see extended DVT prophylaxis in hip replacement again, and prophylaxis of ischemic complication of both of unstable angina and non-Q-wave MI. These are cardiac patients, and you can see inpatient treatment of acute DVT with or without PE, and outpatient treatment of acute DVT without PE. These are clinical evidence that are there. I'll mention them by name in my next slide. In 2000, there was a clinical evidence on DVT prophylaxis in medical patients. And in 2007, recently, treatment of acute STEMI managed medically or with subsequent PCI. So Clexane has really broad clinical evidence and studies. When you are using Clexane, you are sure of the product, that this product has undergone rigorous clinical studies and there is clinical evidence. This is a snapshot of some of the clinical studies that has been done in acutely ill medical patients. You can see Medenox study, orthopedic surgery, you can see Bagvis et al. General surgery, we have a Noxacan study, come back to VTE treatment, Levin et al. And acute coronary syndrome treatment, and stable angina and STEMI. We have Essence, we have TIMI. There are a lot of, this is just a summary of some of the trial, and you can see the relative risk reduction that Clexane offered in most of these clinical evidence. So when you are using Clexane, you are using a product that is evidence-based, and you can confidently give it in this indication. And when you do that, you do the right thing. The rest, Mungu will take care of. Now, back to the subject that we are trying also to discuss, the issue of are all the issue of biological products and their similarity and all that. And the question is, when you are looking at Clexane as a biological molecule, will you say that are all low molecular weight heparin the same? From the presentation of Daktari, I think you already have an answer on this. But this has been mentioned by Daktari that the biological molecule are derived from living cell culture, either from the animal or plant materials, and has very complex structures, very strong process dependent. I am going through this quickly because it's been mentioned. Difficulty to characterize. Their molecular composition is very huge. They are unstable in certain external conditions. And so this product can easily be concluded not to be the same. And I can confidently say that there is nothing equivalent to Clexane. And as I started and trying to reflect you on whether we have biosimilars, because biosimilars meet nearly the same stringent, regulation that has to be approved against the innovator product. But do we have that in Kenya? So the clear answer is that the low molecular weight heparin that we have in this country are not the same. There is nothing that is the same with Clexane. Now, and let's go to the regulatory bodies and low molecular weight heparin. What do they say? And one of the bodies, the regulatory bodies, let me just call it ACCF, and American Heart Association Health Policy Statement on the therapeutic interchange and substitution. And remember, these statements are not based on the copies. These are statements that are based on biosimilars. So as I go through these statements, think about biosimilars, because I don't know what to say about the copies, because you saw a lot of unknown. So we don't know what to say about them. But let me just talk of the biosimilar. And this regulatory body says that the specific use of low molecular weight heparin depends on multiple factors, including patient factors, timing of administration and specific drug availability. They are not interchangeable. Yeah, specific low molecular weight heparin, as well as their generic or so in quotes, they may exhibit assay-based different and immunogenicity. We've seen that, and accordingly, healthcare teams need to be vigilant about their therapeutic interchange in the patient group. So there is a call for vigilance when you are using biosimilars. Please note copies. There is need to be vigilant when you are using them. So you can use them, but with vigilance when you are dealing with biosimilar. And it goes to American College of Chest Physician guidelines, says that low molecular weight heparin are prepared using different polymerization processes. And so they differ to some extent in their pharmacokinetic properties, their anticoagulant property, and their recommended dosing regimen. Therefore, these drugs are not interchangeable, marked on a unit for unit basis. Yeah, so these drugs, you cannot just interchange them. Even if you get the biosimilars we are talking about here and you want to substitute them, then you need to really find out if you can substitute them on a on a unit for unit basis. So meaning that the dosage regimen will be different from one to the other. You have to be sure about that before you substitute them. Then the international consensus statement, they say this: regulatory bodies in Europe and North America, they now consider various low molecular weight heparin to be distinct drug products. They require clinical validation for specific indication for each drug. Therapeutic interchange among these products are not appropriate. So this brings a very important aspect of clinical evidence. So when you are using biosimilars, there is need to have a clinical evidence in that particular therapeutic indication, because this product might be indicated for some use, but are not approved for certain indication. You cannot just say that it's a DVT prophylaxis across all indications. It might be used in cancer patient, but might not be used in cardiac patient for DVT prophylaxis. And to conclude on the regulatory bodies, European Medicine Agency say biosimilar version of low molecular weight need to demonstrate therapeutic equivalence in at least one adequately powered, randomized, double-blind parallel group clinical trial. So they even go to specification on the kind of studies that a biosimilar has to meet to merit its use as compared to the innovator or the the reference biologic product. So these are stringent rules that are biosimilars to go through. So, and not the copies that we have. Now, back to Clexane. Clexane has been demonstrated to have very unique biochemical structure that has been compared to fingerprints. We know that fingerprints, every human being, billions of us who are living in this earth, has distinct fingerprints. This is the molecular or biochemical structure of Clexane. So there's nothing closer to Clexane even here at home. And another thing in terms of excellence, in terms of production, Sanofi ensure that there is excellence, production processes with high standard of quality assurance, and there is a multi-system approach to ensure that the final product is actually meets the standard that is required. Actually, I've read elsewhere that the company goes beyond the regulatory requirements to ensure that the final product meets the standard. And they start from the quality control of the source. As Dr. was presenting, you noted that even the location of production, the change on the structure of the system of production might interfere with the final product. So what Sanofi has done is that there is a particular farm that has a particular location in France where all the material are taken from. And there is periodic audits on these farms to ensure that the animals from where Clexane comes from are standardized. So you cannot have a farm from one end of the country and the other end and come up with the same product. The other thing is that the product can completely be traced to the animal where it comes from, to the final product. There is complete traceability. They can tell this if there is a problem with any batch of with any product, they can trace and see whether was it a problem of how the animal are kept or what they were fed on or anything or whichever condition. So there is a complete traceability in case there is any product. That cannot be said of any copies that we have in this country. Then there is this systematic screening of the animal to ensure that, most animal will have viruses in their body. So there is the viral inactivation. They screen and they do inactivation of the product. And then there's a control and release from a particular distribution location that goes through the world. So we don't have so many plants producing Clexane. It has to be done from one particular source. That is the only way to ensure that the final product meets the threshold of what is intended. So low molecular weight heparin are not the same, as we have seen. Now, just as I try to go into conclusion, I decided to put some injection technique on Clexane because we've gotten a lot of feedback on different position of administration of Clexane. And it is good to note that Clexane should be administered between the left or the right antilateral or posterolateral abdominal wall. And you introduce the needle into a skin fold between the thumb and the forefingers. And you should hold that skin fold throughout the injection time, and there should not be rubbing to minimize bruising of this. And this I've put on a pictorial display here to make it easier to grasp, because we've noted that there's a lot of injection of Clexane on the deltoid muscle, and most patients complain of a lot of pain. I know a lot of you here logged in are in facilities and probably get this kind of feedback. Please advise your nursing teams to give Clexane on the abdominal wall, skin fold. Clexane should be given the way insulin is given. The problem with deltoid muscle is that it's very muscular, and when you put it, you are actually doing intramuscular injection, and it becomes very, very painful, and a lot of patients complain. So please be our advocate, talk to your nursing team so that they can give Clexane on the abdominal wall. And again, this makes it easier, even for patients who are continued on Clexane post-hospital, can easily now, if they are to administer it in the deltoid muscle, you can't do it yourself. But for abdominal fold, one can do it at a seated position comfortably in your house, just the same way insulin is done. Now, just on the dosage, I will go quickly through this because it's a very busy slide. So for prophylaxis of DVT, Clexane is given in medical patients with restricted mobility. You just give 40 milligrams once a day, 6 to 11 days, up to 14 days. But the teaching now is that as long as the risk is perceived, you can always give Clexane. And in unstable angina and STEMI, you give Clexane 1 milligram per kilogram body weight twice daily. And in STEMI patient, you can give 40 milligram IV bolus. The duration is between 2 to 8 days in these indications, but of course, the clinician judgment is always called for. So as long as the risk is perceived, you can always give Clexane. One thing to note is the STEMI patient 40 milligram IV. I just wanted to emphasize that you can give Clexane as an IV formulation in some conditions if need be. And inpatient and outpatient treatment of acute DVT. Let me just emphasize on the issue of outpatient treatment. This is because we are being boxed into the inpatient treatment. I'm not insisting that you must use Clexane as an outpatient, but I can only tell you that Clexane can be used both as an inpatient and outpatient treatment of acute DVT. So when you are using it for treatment, you do 1.5 milligram per kilogram body weight subcutaneous once a day, or the commonly used is 1 milligram per kilogram body weight twice daily. So these are the dosage or administration of Clexane. But in surgery, you can give 40 milligrams subcutaneous 12 hours pre-op, and next dose is 12 to 24 hours post-op. It's always good to note that I know most doctors do not go for the pre-op administration because of fear of bleeding during surgery, but it's good to emphasize that within 12 hours, the coagulation parameters shall have normalized. So a doctor can confidently give Clexane. There are drugs that you wouldn't take a patient to surgery even within a week, but in Clexane, 12 hours is the standard. So post-op, you can do 12 hours or you can do 24 hours, depending on the patient factors. You can make a decision. The clinician can make a decision. And in surgery, we are talking of 7 to 10 days, or as long as the risk is still perceived. But in orthopedic surgery, total hip and total knee replacement, it has been demonstrated that you have to go up to 35 days. The teaching today is that you can continue as long as you perceive the risk. Now, just to summarize in terms of what we have said about Clexane, in terms of quality, we have said the manufacturing process is done in a multidisciplinary approach for quality assurance. There is quality assurance, periodic audit, systematic screening, and viral inactivation. So the quality of Clexane is guaranteed. We've seen a lot of clinical evidence, both from prophylaxis of acutely ill medical patients, and I dare say even critically ill medical patient in ICU should be on Clexane. And prophylaxis in orthopedic and general surgery, DVT treatment, and across acute coronary syndrome spectrum. And we've seen clinical evidence in this. In terms of experience they use, you can agree with me that Clexane is a household product in this country. But I will emphasize that if you go to Europe, if you go to America, I dare even say Russia, I don't know of Russia that much, South Africa and most of the West Africa country, you'll find Clexane. The other name of Clexane is Lovenox. So those of you who have been privileged to be in Europe or America, I'm sure you can resonate with what I'm saying. But just to mention that some of these copies that we have in our country, you will not get them in these regulated markets, but you will always get Clexane. So we say Clexane has proven and predictable protection. And as I end, I want to call upon PSK, and we've talked to, I think the other time we had a meeting, some of you are also members of HOPAC, and this is call to action in terms of biologic molecules. These are molecules that we've seen even from the presentation that they are very sensitive kind of molecules. And it is good for us to ensure that where you saw some of these molecules, where they come from, please let's not treat them as conventional molecules. We've seen even insulin coming from sources that you are not so sure. And sometimes when patients are not controlled, you don't know what they're actually using, because if a guy is going to bring lanterns with a briefcase, then you wonder how was it stored? How was it transported from the source up to this country? For us as Sanofi, even before our agencies are approved to have some of our biological molecules, we are also a household of vaccine, and you guys know that, the insulin and now Clexane. The agency has to be checked. There's reports that are being done. We have a whole quality control pharmacist who ensure and visit and file reports to Sanofi to ensure that even the suppliers have put this product in the in the right temperatures, and it is measured all the time. So these are products that you wouldn't want to source from unknown sources. So when it's come to biological molecule, I call upon PSK as a professional body, and some of you who are members of HOPA, to preach this so that we ensure that for biological molecules, we use the right medication, and we are sure that whatever we are using is coming from the right source and not some of these parallel, we don't know where they come from. Some of the parallel have been in this industry for almost over 15 years. And I can tell you that some of the products we've collected in the market and taken to our to some of the manufacturing sites for studies, it has been revealed that some of them do not even have active ingredient. And they are, our laws allow for parallel importation. There is no quarrel about that. But how do you ensure that whatever you call parallel is a genuine product? And especially when it is a biological molecule, how was the transportation? How is the storing? So this is a call for you guys, and I beg to come to a conclusion and say that Proclexane is a product that you can trust. Thank you, and back to you, Dr. Hezron.
Right, thank you so much, Maurice, for that presentation. I see we have a couple of questions, and we'll just jump right into them. Dr. Imran, are you there?
Yes, I'm here.
All right, thank you. So we'll start with the first question, which is from Dr. Charity, who asks, between biologics or biosimilars and chemically synthesized drugs, which ones have a better safety and efficacy profile?
Okay, that's a good question. I don't think that you can compare the two. Each of them have their own comparators, right? So you're talking about with biologics and biosimilars, you have a different way of assessing the efficacy and safety. With chemical drugs and their generics, you have a different way of assessing by what we call bioequivalence, right? And then as far as now safety and efficacy is concerned, but I wouldn't say one is better than the other. So just in short, they each have their own safety and efficacy profile. So it depends on every drug and how long they've been in the market, right? You can't say that biologicals have a better safety profile than chemical drugs, but you know, of course, the risks are higher when you are now trying to get a biosimilar with a biological product and a biosimilar, because there's so many of it's so complex that the risk of getting a product that is not efficacious enough because it didn't meet certain processes is higher. And so from that perspective, you could say, but you know, if it has a higher risk or it doesn't meet the efficacy profile of the reference product, then the question is, why is it even there? So in that sense, then yes, there's a higher risk in that if you look at it that way.
All right, thank you. The next question is from Dr. Dan, who says she's worried about where this leaves the public sector product selection when the main supplier strictly sticks to generic stocking, even for biologics such as low molecular weight heparin. So how should we objectively deal with this issue as pharmacists whose patients are on limited budgets?
Yeah, it's a very valid question. And I think it brings the bigger question of how are we financing healthcare in the country, and how are we ensuring that all patients, regardless of their ability to pay, are getting the excellent healthcare. And so this is the question of health financing, of universal health coverage. I think it's been belabored a lot, but really we need to be moving towards where we're making sure that our patients are accessing the quality health products, but also at an affordable price, or even free of charge, ideally. But the question is, if KEMSA or is only sticking to generics, then there needs to be a question about what happens to biologicals, because those are a different category of products. So do we need to specify that biological products have a different requirement when it comes to stocking in our main supplies authority? What and do we require that if there's no biological biosimilar, then we need to make sure that they are stocking the reference product. So these are all questions that the regulator and I think PSK as a whole can address, and all of us as members. So yeah.
All right. Thank you for that. Just the last one before we go to Maurice. On the structure, do regulators insist on similar secondary, tertiary, and quaternary structures? And if yes, what would be the justification if the manufacturer is still expected to provide PK and PD data?
Yeah, a very, very good question. So, we talked about, if you remember, we said the FDA uses a stepwise approach, meaning that you have to meet the requirements at every step of the way. So if you don't meet the structural and the functional requirements for that biological, then you won't move to the next step. So they require that, yes, the primary amino acid sequence has to be identical, but then they will also look at your comparisons with the secondary, tertiary, and quaternary structures. Even other things like, of course, we talked about some changes are acceptable, but as a whole, they would look at that. And it also doesn't make sense for you. I think logically speaking, if a product is not similar in all those aspects, and we're not saying identical, but not similar, then you would expect that the pharmacodynamic studies that would be done later, the PK studies that would be done, would not also be identical, or would not be similar. But again, remember, it's stepwise. So you would first do the structural and functional analysis, move then to non-clinical, the animal studies, then go to the PK in humans, then go down to efficacy and safety. So all of those need to be met, but in a stepwise fashion. So I hope I've answered that adequately.
Yes, I think that's very clear. So now the next question goes to Maurice. We have a couple of questions, and the first one is from Dr. Jen Wanjiku, who's asking if Sanofi has a summarized risk assessment tool that can be used by any healthcare worker at a hospital setting.
Okay, I wouldn't say it is a Sanofi tool, but I think there is a lot of VTE. I believe she's talking about VTE risk assessment. And I believe there are several tools that can be used, although we've always advocated for Caprini score. And I think it is very elaborate and well detailed, but there's a lot of other tools. There's Padua score, there's Well score, there are so many. And I think there are facilities that are already using them. So the answer is yes, they are there. They are not Sanofi's, they are scientific tools that have been used for medical management of patients. Okay.
All right, thank you. There's a question again from Dr. Samson Juma, who's asking, can Clexane 60 mg be used prophylactically after total hip or knee replacement surgery?
I think the answer is yes. The clear answer is yes, because basically prophylaxis dose is 40 milligrams. That has been recommended, but clinicians are free to do their judgment. I've been into conference where there are discussion on the issues of weight, and some patients are said to be more weighty, and probably the 40 milligram for prophylaxis might not be adequate. But when you are talking of particularly this kind of surgery, the patients might need to be on them for quite a long time. So you also don't want to go on a very, very high dose. Maybe even if you start with 60, at some point you might want to come back to the 40. Remember, you are doing prophylaxis. You are not doing treatment. So this can be left to a physician judgment. So if you see a doctor prescribing 60 milligram Clexane for prophylaxis, I believe he knows why, and the clear answer is it's possible. Thank you.
All right, thank you so much. We have another question here that could be responded to by both of you. The question is, do we have biologics manufacturers in the country, and what is needed to undertake this? And they also add, this is now helping us to move to more precision and targeted medicine rather than general targeting of viruses and bacteria. Is there a risk for biological weapons being developed for future? Maybe if you have any comments on that, Dr. Imran or Morris.
Let Dr. start.
I don't know about whether we manufacture biologicals. I have not heard of any company, but maybe I stand to be corrected. How do we and what is needed to undertake this? I think, as you saw from the presentation, if you want to manufacture, I would imagine before we even get to biologicals, we would need to manufacture biosimilars, and the requirements are very, very stringent. And so it would be helpful if we could, and I challenge the industrial pharmacists that are here to take us that route. But yes, it is now. It would be helpful in terms of targeted medicine. And then risk for biological weapons, I don't know. I'm not an expert on that, so I cannot comment on that. Morris, maybe you sound more like you know about this.
Not really, Daktari. I wouldn't say that I know if we can manufacture biologics, but I think I can just think or reason logically on something that can be agreeable. That probably what we could be having in the country are the biomimics, the copies. Biologicals, I don't know. I honestly don't know, but I think we could be having these non-comparable biologics. And the issue of weaponize them, okay, for these copies, there are a lot of things unknown about them from Daktari's presentation. So we wouldn't know, but in my own thinking, I think I like using this example. We don't have as much mangoes in this country as we do the mango juice. So definitely what we have are flavors, the mimics of the mangoes. And I can equate maybe probably, I'm just thinking that this is probably what we have. I think we already have this coming from India and other countries. And I think we could be having some, but still the materials are coming from outside. I think what we do here is just putting them together. That is my answer. It's more political. It's not very accurate. You can decide what to think about it.
All right. Thank you so much. There's another follow-up question on that. Dr. Kangogo says the cost of Clexane is a disadvantage to some patients in the community. Is there ways you subsidize its procurement, or are there plans to lower it? And I think just in line with that, someone was asking if, Dr. Samson Juma was asking if Sanofi has access programs for their products.
Well, we have an access program for one of our products, an insulin called Lantus, insulin glargine, long-acting basal insulin. That is there, and I think it has enabled us to reach several counties and several places in this country. You certainly have seen Lantus in your facility in the last few, I think 5 or 6 years. Initially, Lantus was a reserve of Aga Khan, Mata, and the likes of Nairobi Hospital. But right now it's well available. So yes, one of our product launches as an access program. As for the cost of Clexane, the answer to this is this. We have put Clexane through meds, and meds get a subsidized tender price, which is quite accessible in most part of this country. We've also enabled our counties, the government facilities, the likes of, let's say, Kenyatta, MTRH, and also even Coast General Nyeri, to be able to access Clexane from our main suppliers by sending to them an LPO. You just need to do an RFQ that has a logo of your facilities. It has to be proven that this product is going to a government hospital or a faith-based hospital like Tenweka. And when you do RFQ to our agencies, which is Sagifam and Laborex, they will be able to get you the specialized price that is quite accessible to Clexane. But a lot of you here have not exploited this route. So yes, we have two routes. We have meds, and we have you have the ability to ask our primary distributor, Laborex and Sagifam. Just send them a RFQ and they will give you a quotation, which they will have to get from Sanofi head office. Immediately you send this RFQ, is forwarded. And when they get approval for your special price, then they can supply to you. So this is something that can be exploited. Of course, we also do a lot of tender even with the private facilities. And most of the facilities has also been able to get Clexane at reasonable. It might not be the same with the government facility, but reasonable prices. But just to conclude on the issue of cost, sometimes it's good to look at the therapeutic niche of some of these products, and depending on is it a short-term treatment or is it a long-term prophylaxis and all that, because if you are going to use Clexane for prophylaxis on medical patients for 7 to 10 days, I believe it can be manageable. But when you are going to use it for 6 months or so, then you might want to exploit this. So the pharmacists that are logged here can also help us to enable patient access Clexane at this reduced price. And my last comment will be that Clexane might not be at the same equivalent price with the copies, the biomimics, the flavors that we have, but at least you can get it for some reasonable price. So it's my call to you to also enable this patient get Clexane, the original product, by sourcing it from meds or by sending a request, an LPO, to our primary agencies, the Sagifam and Laborex. If you do this, you will help a lot of patients get Clexane at a reasonable price. Thank you.
All right, thank you. Thank you so much for that response, Maurice. I see we're almost running out of time, but then just before we conclude, there's a poll that we have here. I've just shared the poll, so if you can see it on your screen for our participants today, you could just respond to the questions that have been shared. Maybe, Maurice, you can confirm if you can see the poll on your end.
It's visible. It's visible.
Okay, thank you. Thank you, Doc. Yeah, so I can see some responses are coming in. Just on your screen, you should be able to see a poll and you'll be able to respond to the questions. Meanwhile, Maurice and Dr. Imran, I'm seeing some questions on whether you could share the slides.
Sure, I can send it to you and then you can share with members. Yeah.
Okay. All right, thank you.
I also seen a question regarding if someone asked if we can put the chart, the pictorial on administration of Clexane. Maybe Maurice, you want to put that slide up as
yes. Maurice.
On mute.
Maurice, you're on mute.
Okay. Sorry for that, but I could hear you. I'm trying to.
Oh, it's okay.
Share. I think I need to close this poll. It's blocking me to get the tab.
Okay. All right. I think we've gotten a good number of responses. Meanwhile, as you just share your screen.
Is it visible? I'm trying to get the...
Yes, it's visible.
Okay.
So, Dr. Paul, there's the image that is showing the point of administration of Clexane. Maybe, Maurice, you can just briefly just explain again shortly.
Okay. What this is like 2-inch from navel, just the same way you do the insulin. You make a skin fold. This is what's called anterolateral abdominal wall, but you can also do it post-lateral at the back. You can do the thighs. You can do the biceps and all that. But what is important is that you make a skin fold and inject at 90 degrees. The Clexane needle is a very small one. It's not the long one which we were given when we were young at our deltoid muscles. So you make a skin fold, let the whole length of the needle go inside, hold it, release the medication. There is always an airlock. When you do that, that airlock is not meant to be released. It just comes back and then you push the drug. Then you should not rub it. You will note that most subcutaneous most drugs that are administered subcutaneously will always have some kind of, let me say, swelling, because the drug has just gone under the skin. So you allow it to be absorbed slowly. If you bruise it, if you try to rub it, then you cause bruising. So that's why you shouldn't do that. You should just allow it to. Once the drug is all gone, you just release the needle. I think that explained it very well, Doctor.
All right. Thank you so much for that. So
I can pull it now.
Yes.
My goodness.
So I've just shared the responses for those who participated. On question 1, which one of the following best defines a biosimilar? And option 3 was the right answer, which is a highly similar and interchangeable version. A highly similar and interchangeable version of an already approved biologic drug. And the second one, which category of biologics often includes antibodies that target specific diseases or conditions? And the answer there is monoclonal antibodies, of which most of you got. And then the next question is on biosubstitution is a term used to describe the process of switching a patient from a reference biologic to a biosimilar. And the answer there is true. And then the fourth one is interchangeability of biosimilars with reference biologics is determined on a case-by-case basis for each patient. And the answer there is true. Then finally, the last question was on name one biological product from Sanofi, of which most of you have responded on the chat. I think, Maurice, you can approve that it's true. Clexane is one of them.
Yes.
All right. So thank you so much. At this point, I'd like to just welcome Dr. Imran to give just a parting shot before we get into the closing remarks.
Thank you so much. I think I don't have much to add. I believe I'm hopeful that this was an informative presentation for those who attended. And I would like to extend my thanks to PSK for organizing and for Sanofi for their sponsorship. I believe we are, I as well have been enlightened on a lot of these issues. So I hope everybody who participated benefited, and I look forward to future webinars organized by PSK. Thank you.
Right. Thank you so much, Dr. Imran. Maurice, you could give your...
Yes. Thank you, Dr. Just to appreciate all the participants. We couldn't be here without you. And we appreciate all your support. I think I've said a lot, and I've said the things that I don't want to repeat again. So my only appreciation to you guys for coming on these webinars, and we look forward to do similar webinars with you in the future. Thank you, Dr.
All right. Thank you so much, Maurice, and for the support from Sanofi. Dr. Imran, thank you so much for the insights you've shared with us today. So as we close, I just want to briefly take you through a few upcoming events that you should take note of. First of all, this Thursday on 2nd of November at 7 p.m., we'll be having a meet and greet for pharmacists in regulatory affairs and pharmacovigilance. So if you're a pharmacist in these areas, kindly make sure you join that event that we'll be having soon. Be on the lookout in the WhatsApp groups. We'll be sharing the link so that you can register for that session. The next activity is happening this weekend. This will be an outdoor activity. We'll be having a Zumba dance at the Arboretum starting at exactly 9 a.m. So for those who are coming, kindly ensure that you come in early. We'll be having it for 2 hours from 9 a.m. to 11 a.m. at Nairobi Arboretum. Then the other thing is, as you know, we're having the 44th annual conference, scientific conference coming up soon. So we've made a call for a logo that is going to be representing, PSK will be turning 60 years. So we've made a call. We want you to participate in creating a logo that will be used to market the conference next year. And it will be in partnership with the International Society for Pharmacoepidemiology. So kindly participate. For the winners, they'll be announced on 17th, where we'll be having a dinner and dance at Safari Park Hotel. So kindly for the dinner as well, make sure you register early. Our early bird ends on 5th of November. So book your slots early, and we'll see you at the dinner. So thank you so much, and thank you for joining us today. For our sponsors, Sanofi and the team, Maurice, thank you so much. Dr. Imran, thank you so much for sharing the insights with us as well. Have a good evening.
Thank you. Bye.
Thank you. Good evening.
Right. Good evening.